Hi, welcome back. So this video that I'm gonna look at is a fully automated microbiology laboratory for the University of Rochester Health System. When I say fully automated, I mean that it has a ton of instrumentation that you don't find in your typical hospital laboratory. This is a core laboratory. I'm pretty sure they don't say it straight out, but I'm pretty sure it's a core laboratory where they do a ton of testing for all sorts of things, even like virology and serology, pretty intensely. So it's crazy how much stuff that they do. So I'll go through kind of what I see and my experience working with some of the instrumentation there. A lot of the instrumentation I haven't worked with before, so it's cool. It's always cool to see new stuff or to see different things. And I hope you enjoy what this looks like. Thanks.
Hi, I'm Andrea Howard, a postdoctoral fellow in the Medical and Public Health Microbiology program here at the University of Rochester Medical Center. I will be your tour guide in this video, and on behalf of Dr. Dwight Hardy, our laboratory director, and our laboratory staff, welcome to UR Medicine's Clinical Microbiology Laboratory.
This lab tour video will highlight diagnostic testing that is performed in a clinical microbiology laboratory. Clinical microbiology laboratories play an important role in patient care by detecting, isolating, and identifying microorganisms that cause infections. The field of clinical microbiology is multifaceted and multidisciplinary. Here at UR Medicine, we perform bacteriology, immunology, mycobacteriology, mycology, parasitology, serology, and virology testing to assist clinicians in the diagnosis and management of patients with infectious diseases and immunological disorders. The process of identifying infectious organisms begins with a sample from a patient being received in the laboratory. This is our specimen receiving area, where specimens are received from various hospitals and affiliates that.
So it looks like this is a core laboratory. What that means is it's not attached physically to, or at least I don't think it is, attached to a hospital. It's an independent site all on its own, specifically designed for laboratory processing and laboratory results. This is similar to things like Quest, LabCorp. What's the other one? I can't remember the other one. But so what that means is you'll have all of the hospitals, outpatient facilities, all at their own sites, and then you'll have everything will be sent to that core laboratory in different locations. So you'll have a lot of transportation moving to and from each hospital to the core laboratory for specimen processing and testing and everything like that. So at least that's what I think this looks like, because usually hospital laboratories are not this big. For example, where I live, UPMC and Allegheny Health Network, they have their own core laboratories because they have, between them, there's like 10 different hospitals. And instead of having a laboratory in each hospital, it's more efficient to just have one core building designed specifically for laboratory testing. And then they all send their specimens to their designated core laboratories.
Use our services are logged into our laboratory information system and are sorted by sample type for the respective lab benches to perform diagnostic testing. This is the specimen setup and stat test area. A stat test is a test with a quick turnaround time, generally less than 1 hour, as results are needed urgently for patient care. In this area, some of the tests we perform include rapid antigen detection assays for malaria, Legionella, and Streptococcus pneumonia. Here we observe a technologist performing a lateral flow assay to detect Clostridioides difficile in a patient's stool. In the stat lab, we also perform rapid PCR using the Cepheid system. This detects nucleic acid targets for bacterial and viral pathogens, including Microbacteria tuberculosis, Staph aureus, Strep pyogenes, COVID, flu, and RSV.
So the Cepheid, that little cartridge there in the center here, this really changed the game around when COVID came out. I think they were the first to develop a rapid PCR test for COVID-19. When I first started out, these were in high demand, and we would run out of them all of the time. A lot of emergency room protocols at the time, I'm not sure if it's still the case, but at least where I worked at the time, this was in 2020, 2021, required a patient to have COVID testing prior to hospital admission. So that means every patient that came in that was going to be admitted needed a COVID-19 test. The COVID test takes about 50 minutes, roughly an hour, once you include processing time. That's a lot of reason that the waiting rooms were like backed up, or the emergency rooms were backed up, because they were waiting for these COVID tests before they could send them to the floors. But that little cartridge for PCR testing really changed pretty much everything. Like they were saying, it can do MRSA testing. It can do it can do some. I can't remember if they do blood culture IDs or not, but it can do C. diff testing. That's pretty much a staple now in most hospitals. COVID, flu, RSV, called multiplex testing or threeplex testing. And it can do a whole bunch of other stuff. Strep A testing, CTNG testing, for urine. It can do a whole bunch of really cool things. Yeah.
Well as detects markers for antibiotic resistance, including methicillin-resistant Staph aureus, rifampin-resistant tuberculosis, and carbapenem-resistant enterobacteriales. Here, a technologist is setting up a swab to detect Staph aureus colonization in a patient. The PCR reaction is contained within the test cartridge. Once the patient sample is added, the cartridge is loaded onto the machine. The Cepheid system is a on-demand random access.
So this thing is massive. One of the places I worked with had one. I'm not sure if it was this big, but it had the automated arm. And so each of these little channels here is an individual test cartridge that'll be inserted. It can run a bunch at the same time. That's why it makes it so efficient. There's no manual loading or unloading. That saves on lab personnel time. Results are automated and crossed over to the LIS system and then resulted to the doctors automatically. So it's also very expensive. Several $100,000, if not $1 million. This is the big one. They make smaller ones. I think the smallest one they make is either a 4 or 8 capacity one. Obviously no automated arm for those ones. Those ones you just load and unload yourself. I think the one that we have at the hospital I work now, I think is 20. But this has a huge capacity. Again, running all the tests at the same time. Pretty amazing.
A system that allows multiple tests to be run at the same time as needed, providing rapid results. In our bacteriology lab, we utilize the Keystra automated microbiology system. This system automates traditional microbiology, which is the growing of bacteria out on petri dishes, and the steps that used to be done manually, specimen processing and culture inoculation, can now be done automatically by these machines. Specimens are processed step by step from inoculation of agar plates.
So this is an automated specimen plating system. Each of these plates is a different patient, or because that looks like blood, McConkey, and chocolate, so that may be one patient. But it'll plate the specimen for you, and then it'll along a assembly line. We'll send it to the different incubation places as needed, whether it's O2, CO2, whatever. These things are expensive. I think, if I remember correctly, because I remember looking, I think this is the Kestra system from BD. That whole automated system costs about $1.2 million. That's a lot of money for hospitals and everything for testing. But it cuts down on personnel time. It cuts down on a lot of stuff. The ones that we used at my old job, I think we used the Cobus Wasp. That we used that only for urine cultures. We had 3 of them. There was no assembly line or anything. It was a self-contained like unit. But that processed urine cultures, and it would just, we had a lot of issues with it. That's why they had multiple of them, because they had a lot of issues with one pretty much every day. It was kind of rare to have all 3 up and running at the same time. But they can do a ton very quickly. And that's really important, because when you have where I work now, at the little hospital I work now, you have to take out the plates. Depending on what plates you need, you may have to warm them up. You have to take them out in advance, lay them out under the hood, put the stickers on each one, get your specimen, swab the specimen on the plate, streak for isolation for each plate. Sometimes you have like 6 plates that you got to do. That's a lot of lab tech time to spend on one specimen. So this cuts all of that down, so that the tech can go off and do other things. So it's a pretty impressive system. I wonder if this thing ever breaks down, what they do, because this is obviously a high volume operation, similar to where I was working before, but probably more so, especially with things like wound cultures, blood cultures, and stuff like that. It's pretty impressive.
To colony isolation, to incubation, and transfer throughout the system by conveyor belt. High volume samples such as urines, bloods, and e-swabs are completely processed by the Keystra. Here we observe a urine specimen being set up for culture. Specimens and auger plates are barcoded so they can be tracked throughout the system. First, the specimen is scanned for the selection of the appropriate auger plate for growth, then vortex, and an aliquot is removed for inoculation onto the auger plate. The machine adds magnetic beads to the plate to allow for a standardized culture inoculation.
Oh, that's cool.
We can see here the magnetic beads streaking culture inoculum across the auger plate to allow for growth of isolated colonies. Other specimen types are set up semi-automatically. Various auger plates may be used to optimize growth of the potential pathogens within the sample. Each auger plate contains the nutrients that different bacteria need to grow, and the type of nutrient auger that the specimen is cultured on depends on the body site that the specimen is collected from. Here we observe a technologist inoculating various patient samples onto auger plates. Along the Keystra work line, we have reader and worker benches. Technologists at the reader benches evaluate the growth of microorganisms on auger plates following overnight incubation.
So all those plates that you saw, you still have to have a physical lab tech, a person, read those plates, interpret those plates, and look at what is potentially growing, what they need to work up, what they don't. Although I like the bead thing. The bead thing is really cool. The wasp that we use would use disposable loops to swap the plates. But I like the bead thing. And you saw the way that they were. Hold on, let me go back a little bit. So if you can see here, it looks like it takes pictures, which is really cool. And it looks like the quadrants. Looks like it does an okay job. This one looks really good. But it looks like it takes pictures of the plates as well, so you can observe them in the middle of incubation time or whatever. That's pretty cool. It looks like a streak method. It looks like it goes starts here, goes down, boom, goes down, goes back up, goes down. That's interesting. Huh.
For work line, we have reader and worker benches. Technologists at the reader benches evaluate the growth of microorganisms on auger plates following overnight incubation. Technologists can look at plates and determine by colony morphology if it is something that would normally grow from the particular body site, or if it's something that requires further workup, something we may need to identify as a cause of infection in a patient. Technologists at worker benches will perform workup of suspicious colonies. For bacterial identification, The isolated colony is transferred to a target plate and identified by mass spectrometry. The mass spec process develops a protein profile.
Okay, so she's talking about MALDI-TOF. That's what this plate down here is. That's your target plate for MALDI. It looks like a pretty big system. Looking at the number of targets that are on that plate. So yeah, so each of these is probably a different patient. That looks like typical urine culture. That's a split McConkey and blood auger plate. So it looks like he's doing urine cultures, and setting up his IDs for the day.
File of the isolated colony. This profile is then matched against a reference database to allow identification of the bacteria species.
One of them, I'm assuming.
Antimicrobial susceptibility testing, or AST, is performed to guide clinicians in choosing the right antibiotic for a particular infection. This test provides information about the efficiency of
So that's the BD Phoenix, Beckett Dickinson. I've only worked at one place that uses that. I've used it kind of during my initial training. I was the place that I worked where they had this instrument. I was doing specimen processing, so I wasn't involved in any of the MIC testing or anything like that. But it looks like MIC style testing. I haven't heard a ton of good things about the Phoenix. It's a pretty big instrument, and you need a lot of space in order to use it, but I think it has a pretty decent capacity in terms of how many things you can set up. So each of these cartridges is a different patient. So he's working at about 4 patients at a time right there. So it looks like a pretty decent capacity. I'm not, like I said, I'm not sure about the reliability of it. I assume it's pretty reliable if an operation this big is using something like that. So that's pretty interesting.
Microbial agents in treating bacterial and fungal infections. We perform AST on gram-positive and gram-negative bacteria using the automated Phoenix AST system. Panels, which contain various antimicrobial agents, are inoculated with a standardized inoculum of an organism and incubated in the instrument to detect bacterial growth in the presence of antibiotics. In the MIC's lab, we perform manual AST methods, including broth microdilution and disk diffusion assays.
So this is also MIC, but okay, so what's the difference? The difference between that machine and this type of testing is selection of antibiotics. How do I want to break this down? Okay. So the way that it works in most hospitals, or most hospital systems, is you have the pathologist and infectious disease doctors work together to determine which antibiotics you will run for which specific organisms. Now, there are guidelines from different organizations like CLSI, that's a big one, that continuously publish guidelines for which antibiotics to work with for which specific organism. For example, antibiotics that you test for with gram-positive organisms are going to be different than ones you test for gram-negative organisms. Because some antibiotics are specific for gram-positive organisms and antibiotics are specific for gram-negative organisms. You have some that overlap, but that's a different thing. But so we're not running every antibiotic known to man for each individual organism. That would be way too much time. That would be way too many resources, and it just wouldn't make sense. So with those published guidelines, infectious disease doctors and pathologists for each hospitals will look at them and say, okay, we like this, we'll do this. They may make a couple changes, because it's not like a federal law or anything like that. But they all pretty much do the same. They all pretty much run the same antibiotics with a couple differences here and there, depending on your population. You may have a population where you work where you see a lot more Staph aureus than you see Enterococcus in a urine culture. Who knows? Or in a wound culture, you may see more Group A than Group B or something like that. That's all pathologists and ID stuff. They have to keep that in mind. That doesn't have anything to do with the lab techs. We just do the testing. But a doctor may want a specific antibiotic for a specific organism that isn't run on the typical panel. For example, daptomycin. Daptomycin is a antibiotic that is typically prescribed for Enterococcus infections. But it's not run with every gram-positive organism. So you have to do a separate test to determine the MIC or the sensitivity for daptomycin because it's not run on the typical panel. So that's what this young tech, or not young, well, that's what this tech is doing here. She's probably doing specific antibiotics for specific organisms that aren't run on the typical gram-positive or gram-negative panels. I hope I explained that well. I don't know. We'll see.
Assays are performed on organisms where automated panels are not available, such as yeast, mycobacteria, and anaerobes, or for certain drug-bug combinations as requested by physicians. Here, a technologist is reading a broth microdilution plate and recording the MIC, the lowest concentration of the antibiotic that inhibits bacterial growth, to determine the sensitivity of the bacteria isolate to a particular antibiotic. Antimicrobial susceptibility testing is one of the ways the clinical microbiology lab plays a critical role in patient care. AST results are used to develop antibiograms which summarize susceptibility patterns of common isolates within a particular hospital and contributes to antimicrobial stewardship initiatives. We process stool specimens for the detection of gastrointestinal pathogens using the BD-MAX system. This multiplex PCR system can detect and differentiate between viral, bacterial, and parasitic causes of infectious diarrhea. This test allows for rapid diagnosis of infections caused by bacteria including Salmonella, Shigella, and Campylobacter, viruses including norovirus, adenovirus, and astrovirus, and parasites including Giardia, Cryptosporidium, and Entamoeba histolytica. In certain cases, stool specimens are submitted to parasitology for ova and parasite examination, which includes stool concentration and microscopic examination of iodine-stained wet moths and trichrome stains for eggs, worms, and larvae. Certain infectious diseases and autoimmune diseases are...
So parasitology is it takes a lot of tech time. You can see you have to stain it. So that's probably the trichrome stain that she's working with. That parasitology is basically, so you have some PCR testing. They mentioned earlier they use an instrument to look for something called Entamoeba histolytica. That's a parasite. You have some PCR testing available for certain parasites, but for things like tapeworms, you really need to look at a stained slide of the stool specimen in order to look for eggs, proglottids, things like that. And that's a lot of tech time. Place I worked before, we had, dozens and dozens and dozens of what are called ova and parasite tests for different patients. And, it's a lot of tech time. It's a lot of work. It's looking at a microscope 8 hours a day. And it's a lot of, it's a ton of work. It's interesting when you get cool stuff, but a lot of time it's pretty benign or pretty typical stuff.
And microscopic examination of iodine-stained wet moths and trichrome stains for eggs, worms, and larvae. Certain infectious diseases and autoimmune diseases are diagnosed using serological and immunological methods. Blood and serum samples are processed for detection of antibodies or antigens to particular pathogens. The Architect system, an
Oh, nice, the
automated
Architect.
immunoassay analyzer, is used to detect antibodies to syphilis, hepatitis A, B, and C viruses, and antigen detection of hepatitis B virus. This instrument is also used for fourth-generation HIV testing, a combination antibody and antigen assay that allows for early detection of HIV infection. One of the high-volume tests performed in our laboratory is allergy testing, which is done using the Phadia immunocap assay.
Oh, that's interesting.
This assay detects and quantifies IgE antibody levels in patient serum to specific allergens, including grass, nuts, dust, insects, and venom. The Bioplex instrument is used for diagnosis of autoimmune diseases.
Ooh, the Bioplex.
This multiplex immunoassay processes serum samples to detect biomarkers for diseases such as lupus, rheumatoid arthritis, and celiac disease.
Viral loads for
Look,
HIV.
Bioplex is pretty cool. Bioplex uses something called immunofluorescence to detect various antibodies of different potential diseases, including things like Lyme disease. So what it'll do is you'll have your serum, and then it'll take the serum, it'll throw in magnetic beads that are coated with a fluorescent dye. And what'll happen is those antibodies will attach to those magnetic beads. If the antibodies for that potential pathogen are present in the serum, they'll attach to the magnetic beads. And then what it'll do is it'll magnetize all of those beads against one side of the tube, and then it'll shine a light through it. And if there's fluorescence, once it shines the light through it, there's a detector on the other side that'll detect any fluorescence from those beads. And then if you have fluorescence, that means you are positive for those antibodies, which means you are possibly positive for that pathogen. It's pretty cool. They used to do it by hand. I don't know how they would do it by hand, but the Bioplex is a pretty cool instrument. It does a lot of the way that it does it is pretty cool.
Lupus, rheumatoid arthritis, and celiac disease.
Viral loads for HIV, hepatitis B, and EBV can be measured in patient serum using the Cobas 8800 to monitor the response to antiviral therapy.
In microbacteriology,
Okay. So what's the difference between the Cobas COVID testing and testing from the Cepheid, those little cartridges that we saw earlier? Difference is quantitation. So what the Cepheid PCR test will tell you is simply positive or negative. Is it there or is it not there? Boom. What this does is once you have a positive COVID test or whatever positive test for something like HIV or something like that will tell you, those will give you positive, negative tests. Then once they start therapy for different types of viruses for antiviral or retroviral therapy, then they need to determine, okay, how much of the virus have we eliminated or is our therapy working, right? In order to do that, you need an actual number to go off of to determine if that number is getting lower or if that number is getting higher. If the number is going higher, then obviously the therapy isn't working. If it's getting lower, then obviously it's working. So in order to do that quantitatively, you have to do what's called qPCR, quantitative polymerase chain reaction. That's what the Cobas does. It's a little bit different. I can't remember specifically how it's different, but what I know is it'll chuck out a number instead of a positive or negative. So it'll give you an actual viral load, how much virus per however many milliliters of blood that you've been given. So it's much more detailed, and it gives you, that's more for treatment monitoring than it is for initial diagnosis.
To monitor the response to antiviral therapy.
in microbacteriology, we process patient samples for culture-based detection of microbacteria, including Mycobacterium tuberculosis. A blood sample can also be used to diagnose TB infection via the quantiferin assay, which detects immune responses to TB-specific antigens. STI testing is performed on the Panther system. Urine specimens, vaginal, rectal, and throat swabs are processed for nucleic acid detection of sexually transmitted pathogens, including gonorrhea, chlamydia, Trichomonas vaginalis, and Mycoplasma genitalium. In mycology, patient samples are processed for detection of fungal infections caused by yeast and molds. Here, a technologist is performing a tape prep to identify mold from a nail specimen. Molds are identified macroscopically by color and appearance of growth on agar plates and by microscopic examination of fungal structures.
So again, mycology is another one of those similar to parasitology where it's a lot of tech time. Also, mycology takes a lot of time. You have to wait. I think we keep our fungus cultures, I think, for 3 weeks. And we may read them once a week or something like that. I can't remember. I don't do much mycology yet. Someday, hopefully. But so the way that reading my mycology plates works is you're not just looking at the top side, so like bacteriology. You're not just looking at one side of the plate. You're looking at the reverse side of the plate as well. And then what you typically do is take a piece of tape. In order to look at it under the microscope, you take a piece of tape, gently press it on top of it, transfer it to a slide, and then you will have a drop of a dye called lactophenol blue, which is a very specific dye for fungus and mold. And then you'll put a glass slip over that, and then that's how you can read it under the microscope. Again, a lot of tech time. Some PCR testing exists for mycology specimens, not a ton. Well, when I say PCR testing, I mean that like hospitals can use. Obviously, you can do PCR with pretty much anything, depending on what you're looking for. But the type of PCR testing that's done for molds is manual method. So no real instrumentation, no big heavy machinery, a lot of pipetting and stuff like that. Things that you would see more in research laboratories or reference laboratories. Nothing that you would see in a typical hospital-based or hospital system core laboratory.
And by microscopic examination of fungal structures. This is an automated, high-throughput molecular diagnostic system in which up to 376 samples can be processed per batch, which takes about 3 hours. The system utilizes 2 modules for specimen processing. First, 308 weeks, and by microscopic examination of fungal structures. We perform COVID-19 testing using the Amplitude system.
Okay.
This is an automated, high-throughput molecular diagnostic system in which up to 376 samples can be processed per batch, which takes about 3 hours. The system utilizes 2 modules for specimen processing. First, samples are transferred from tubes to plates by a liquid handler system. Then, by use of a robot arm, the plates containing patient specimens are processed for RNA extraction and PCR analysis to detect gene targets of the SARS-CoV-2 virus. The professional career fields that make up the laboratory staff include technicians with 2-year associate degrees, technologists with 4-year bachelor degrees, and doctoral level directors with specific postdoctoral training. Clinical microbiologists also serve as educators and consultants. Our technologists train medical technology students, postdoctoral fellows, and pathology residents as part of their clinical microbiology rotations. This bench-level training is essential for preparing medical technologists, doctoral-level clinicians, and scientists to join clinical microbiology and public health labs in the future.
Wow. Okay. So that was University of Rochester fully automated lab. It had some pretty crazy stuff in it. You could see a lot of the blood culture machinery that they had. You could see, their one MALDI-TOF. I'm assuming that they have more than one, based on just the volume that they put in. So it's really important to remember that that's a high volume lab. And when I say high volume, I'm talking thousands of specimens a day. The lab I worked before was a high volume lab. In just urine cultures only, we would do about 2,000 a day. So imagine doing something like that every single day. Obviously, not one person doing all of that. You have more than one person. But, sorry, that's Scout. But yeah, it's crazy to see how you can just organize all of that stuff. Walking into a lab like that, my mind would get so overloaded and so overwhelmed. It's pretty insane. But hope you enjoyed that. Again, feel free to comment on kind of whatever, and I will see you later for the next one.